So if there is question regarding draw the diagram of the microanatomy of this structure, then you have to only draw a neat diagram, label diagram of the histology. And when there is short note on histology of particular organ, then you have to just give a little background introduction, where it is located, what is the function of this organ. You should know the general part, and then you have to write down 3 to 4 characteristic features, जो key features है, how you will recognize that this is the slide of this particular. तो इसके जो key features होते हैं हर एक organ या फिर टिश्यू के अंदर उसका पर्टिकुलर फीचर होगा, टाइप ऑफ एपिथेलियम के पर्टिकुलर फीचर्स होते हैं, वो आपको लिखने हैं, आइडेंटिफिकेशन इंपॉर्टेंट फीचर्स. ठीक है. तो अभी हमने एक जनरल इंट्रोडक्शन पढ़ा है, तो ये जनरल इंट्रोडक्शन आपको पता होना चाहिए. प्रैक्टिकल, अब ये तो थ्योरी की बात हो गई कि आप थोड़ा सा उसका जनरल पार्ट देंगे, बैकग्राउंड इंट्रोडक्शन देंगे, एंड देन यू विल राइट 3 to 4 इंपॉर्टेंट कैरेक्टरिस्टिक फीचर्स एंड ड्रॉ दी डायग्राम. देन व्हेन इन प्रैक्टिकल डायग्राम ड्रॉ तो करना नहीं आता है आपको, ठीक है, पर आपको क्या है कि उसके नाम लिखा हुआ है. वी हैवंट हिडन द स्लाइड, नेम ऑफ द स्लाइड्स, ओके, देयर इज नेम ऑलरेडी रिटन देयर, but the problem is you will write the name because it is already being shown, but you don't know how to identify the key features, what are the key features. So in this class, if we start revising the general features, तो आप क्या एक concentration on the particular topic which is histology, वो जो है चला जाता है कंसंट्रेशन. तो हम थोड़ा सा शॉर्ट में जनरल एक बार रिवाइज करके देन वी विल ओनली कंसंट्रेट ऑन द हिस्टोलॉजी ऑफ दीज ऑर्गन, और ये एग्जाम्स में आता ही है. So lymphatic system, as we discussed, is a drainage accessory to the venous system. Then what type of the particle? Larger particle that escape into tissue fluid can only be removed by the lymphatic system. So it's an additional system, addition to the venous system. So larger particles are drained by the lymphatic system. Finally, they will drain into the larger veins. So what is the function? First is it reabsorbs excessive interstitial fluid, returns it to the venous circulation, maintains the blood volume, prevent interstitial fluid level from rising out of control. This is its primary function. Then it transports the dietary lipids. As we have discussed, that there are small lymphatic vessels which are present in the small intestine, which are known as lectins, which absorbs the fat globules. Then we have lymphocyte development, and also it provides immunity. So now, whenever there is a slide of this lymphoid organ, you should know the background story. What is immunity? What are the types of immunity? What are the cells of immune system? So now this is your responsibility to read about these, and it is also being covered in your physiology part. So lymphatic system provide protection to individuals by imparting immunity. It helps in the differentiation of the lymphocytes and maturation of the lymphocytes. So what are the components of lymphatic system? Lymph, which is the interstitial fluid, lymphatic vessels which start with the capillaries, then they will form vessels, then trunks, and then duct which drains into the veins. Then lymphoid organs, as we have discussed, there are two types, primary and secondary. And the cells of the lymphoid system, which are majorly lymphocytes. So this classification you all must know. ठीक है, ये background story तो आप journal में पढ़ेंगे ही पढ़ेंगे. Now starting with this, lymphatic organs are divided into two, primary organs and secondary organs. Suppose there is a slide of thymus. The first question will be asked that what type of organ it is. Then you must know that it is primary lymphoid organ. Then the secondary lymphoid organs are lymph nodes, spleen, tonsils, and lymphatic nodules which are present as associated with the mucosa. So primary lymphoid organs are also called central lymphoid organs. They are also known as central lymphoid organs. These organs produce immune lymphocytes from stem cells or immature cells. For example, thymus and bone marrow. So this is an MCQ question. If there will be one option out of these two, and you have to recognize, you have to pick one, identify the primary lymphoid or central lymphoid organ. What is the function of this? They produce immune cells by the differentiation from the stem cells. Secondary lymphoid organs, these organs provide exposure to the foreign antigen and convert immature lymphocytes into active lymphocytes. So maturation of lymphocytes takes place where? In the secondary lymphoid. Now this secondary lymphoid organ is again of two types, diffuse lymphoid tissue and dense lymphoid tissue. ठीक है, जहाँ पे diffuse है, it is not associated with the capsule. There is no capsule, there is no outer covering, and the dense lymphoid tissues जो हैं वो सारे हो जाएंगे lymph node, spleen, जो हम पढ़ेंगे thymus, they are dense lymphoid tissues. Diffuse lymphoid tissues में क्या आएगा? Lymphocytes are distributed throughout the mucosa like in GIT, and they are associated with the mucosa. That is why they are known as MALT, mucosa associated lymphoid tissue. Diffuse lymphoid tissue is not enclosed with the capsules. They are not enclosed in the capsule. So example of diffuse lymphoid tissue क्या हो जाएगा? MALT, which is mucosa associated lymphoid tissue. These are not enclosed by capsule. Dense lymphoid tissue, it is enclosed in the well-defined capsule. Identification feature, it includes lymphoid organs such as lymph nodes, spleen, thymus and tonsils. Cells of lymphoid system. Now what are the cells of lymphatic system? Anyone? Mainly lymphocytes and supporting cells in the form of reticular cells, epithelio-reticular cells. These will provide the structural framework to the lymphoid organ. And some other cells of the immunity system are also there. For example, macrophages, neutrophils, basophils, dendritic cells and so on. So major component of lymphoid system is lymphocytes, and other immune cells are also present. Along with this, the structural framework is supporting cells are reticular cells and epithelio-reticular cells. So what are the cells? We have divided them into following the major lymphocytes: T cells, B cells and natural killer cells. You all are aware of these. T cells are the cells which provide cell mediated immunity, and B cell immunity is basically how many types? What are the types? Innate and acquired. Then innate is by birth, and acquired is again active and passive. Yes. So cell mediated means mediated by the cells, killer T cells. So these are of following types. Now these T cells are divided into memory T cells. जब भी कोई एंटीजन आता है तो ये memory T cells क्या करेंगे? अदर T cells को एक्टिवेट करें. Helper T cells again they express CD4 antigens. Now जी CD का मतलब क्या है? CD सरफेस पे मॉलिक्यूल, CD का मतलब क्या है? क्लस्टर ऑफ डिफरेंशिएशन. उनके बेसिस पे डिफरेंशिएट होते हैं इनटू T सेल्स, B सेल्स, नेचुरल किलर सेल्स. तो इसके सरफेस पे CD4 एक्सप्रेसेस CD4 ग्लाइकोप्रोटीन. देन CD8 एक्सप्रेसेस CD8 ग्लाइकोप्रोटीन. ये थोड़ा डिटेलिंग में, इतना डिटेलिंग में आपको नहीं जाना है. देन दे डिस्ट्रॉयज वायरसेस, इन्फेक्टेड सेल्स एंड कैंसर सेल्स. सप्रेसर T cells क्या करेंगे? दे विल सप्रेस द एक्टिविटी ऑफ द अदर immune cells. So these are the types of cells of cell mediated immunity: memory T cells, T helper cells, cytotoxic cells and suppressor T cells. Then the B cells, they develop in the bone marrow. They produce antibody by the formation of plasma cells. So here again one MCQ question is there, that plasma cells are formed by which lymphocytes? B lymphocytes, and the immunity associated with them will be, what type of immunity it is, which is mediated by the antibody? Humoral. The natural killer cells, they are non-T, non-B cells. They express CD16A, CD56 and CD94 surface molecules. They destroy antibody bound virus infected and cancer cells like T cells. ठीक है. So basically you must know कि what are the cells. Major cells क्या है? Lymphocytes. Then what are the classification of lymphocytes? So 3: T cells, B lymphocytes and natural killer cells. What are the types of the T cells? Which are the killer T cells? So CD8 are the killer T cells. B cells, what they do? They participate in the humoral activity and they produce plasma cells. Then natural killer cells are neither T cells neither B cells. They express CD6 following molecules and they destroy virus or foreign particles. Lymph nodes. What are lymph nodes? So starting with our first topic is histology of lymph nodes. ठीक है, lymph nodes अभी आपने कैडेवर में देखे हैं सबने, grossly, yes. So they are present as nodular masses, round or oval in shape. What will be the basic identification? They are bean shaped. They are convex. They have a convex surface and a concave surface. They are located along the pathways of lymph vessels. So what does they do? What is the function of lymph node? Filtration of the lymph. Then their length varies from 1 to 25 millimeter and they are typically found in clusters. आपने देखे होंगे superficial and deep flow nodes, तो वो 8 to 10 groups में they are present at different sites. They receive lymph from many body regions. So this is the shape of lymph node. It is bean shaped. It has a convex surface and a concave surface. The convex surface receives afferent lymphatic vessels which enters into the lymph node, and then the lymphatic vessels that exit at the hilus are known as efferent lymphatic vessels. So it is a bean shaped organ with afferent vessels entering at the periphery and efferent lymph vessels emerging at the hilus, arranged in groups along the blood vessels, divided into superficial and deep groups. So these are the different types of lymph nodes present at different locations. So our concern is to know about what? Microanatomy of lymph node. अगर हमें स्लाइड मिलती है lymph node की तो हम उसको कैसे पहचानेंगे, ठीक है? Yes, come in. So look at this diagram. Please pay attention. सबसे पहले इस स्लाइड को हम समझ लेते हैं कि अगर स्लाइड हमें लिम्फ नोड की मिल रही है तो इसमें आपको दोनों स्टेन्स दिख रहे हैं, ठीक है, ब्लू स्टेन्स एंड पिंक स्टेन, HNE स्टेन्स, ठीक है. कुछ जगह आपको यहाँ पे डेंस वॉयलेट कलर दिख रहा है और कुछ जगह पे आपको पिंकिश कलर दिख रहा है. हम बहुत जीरो लेवल से इसको पहचानने की कोशिश करते हैं, ठीक है. तो आउटर कवरिंग देख रहे हैं जो हमें इधर लूजली अरेंज दिख रही है, हालांकि दिस इज लेयर ऑफ डेंस कनेक्टिव टिश्यू. ओके, वी हैव सेड दैट इट इज डेंस लिम्फॉइड टिश्यू लिम्फ नोड विच इज कवर्ड बाय द कैप्सूल. तो आउटर मोस्ट कवरिंग क्या हो जाएगी जो हमें पिंक कलर में दिखी है जिसमें बहुत सारी छोटी छोटी माइन्यूट ब्लड वेसल्स हैं, दिस इज कैप्सूल, ठीक है. Now this capsule will send septas here into the parenchyma. Okay, so these septas are known as trabeculae. These septas are known as trabeculae. तो हर जगह यहाँ पे कैप्सूल सेंड करेगा into the parenchyma septas which are known as trabeculae. तो जब आप स्लाइड फोकस करेंगे आप देखेंगे कि ये कैप्सूल्स की तरह है और कुछ डिवाइडेशन दिख रहा है, सेप्टास दिख रहे हैं, तो मे बी ये लिम्फॉइड सेल्स हो सकते हैं, लिम्फॉइड की स्लाइड हो सकती है. देन नेक्स्ट स्टेप इज टू recognize the cluster of lymphocytes which are circularly arranged here adjacent to the septas, ठीक है, and here the outer layer is densely arranged. These are the mature lymphocytes. And here in the center there is lightly stained region which is known as germinal center. So here the immature lymphocytes are differentiating and maturing, and this zone is the mature zone of the lymphocyte. So germinal center में क्या हो रहा है? Differentiation and maturation of proliferation of the lymphocytes, and इसकी outer layer पे क्या होगा? Densely arranged lymphocytes. बेसिकली इस लेयर्स में बी लिम्फोसाइट्स होते हैं. नीचे जाके सो दिस पार्ट, दिस डेंसली पैक्ड रीजन ऑफ लिम्फोसाइट इज द कॉर्टेक्स पार्ट ऑफ द लिम्फ नोड. जैसे जैसे आप अंदर की तरफ जाएंगे आपको यहाँ पे लूजली अरेंज्ड लिम्फोसाइट्स दिखेंगे, और ये जो लिम्फोसाइट्स हैं ये कॉर्ड्स की फॉर्म में ज्यादा है रादर देन दे आर कॉम्पैक्टली अरेंज्ड इन अ सर्कुलर फैशन. So these are the medullary cords. This is the medulla region. Okay, so these are the medullary cords means now these lymphocytes are linearly arranged in the form of cords. Now again here B cells are there, macrophages and the plasma cells are there. Then some blood vessels are also visible. So how many layers are there in the lymph node? There is a cortex, which is the outer one, and the inner one is medulla. Now this cortex, it has lymphatic nodule. This lymphatic nodule in the center has a lightly stained region which is known as germinal center. There are no densely packed cells, that is why it is lightly stained. And basically what is happening here, maturation and proliferation of the lymphocytes. Mature lymphocytes are present in dense fashion surrounding the germinal center. Now this cortex part is again divided into this nodular cortex, which has this lymphatic nodule, and the paracortex, which is present adjacent to this medullary cords. This region basically consists of T lymphocytes. Okay. So there is cortex. Cortex part had medullary cortex, which has lymphatic nodule. Key feature is germinal center. Then medullary cords, linear fashion arrangement of lymphocytes. Yes. Then other than this medulla and cortex, there are sinuses. There are sinuses which are present here. So sinus just below this capsule part, this is subcapsular sinuses. Now I said there is afferent vessel and there is efferent lymph vessel. Afferent जो कि lymph लेके आएगा towards the lymph node. So this afferent opens here in the subcapsular sinus. Now this subcapsular sinus is connected to the medullary sinus through trabecular sinus. ये ट्रेबिक्यूलर के आसपास लाइटली स्टेन्ड रीजन दिख रहा है, ये वाला लीनियर फैशन में. दिस इज ट्रेबिक्यूलर साइनस विच इज प्रेजेंट एडजेसेंट टू द ट्रेबिक्यूलर. नाउ दिस ट्रेबिक्यूलर साइनस इट कनेक्ट्स सब कैप्सुलर साइनस टू द मेडुलरी साइनस. हियर ये जो आपको पूरा स्टेन दिख रहा है ब्लू कलर में, ये क्या है? मेडुलरी लिम्फ नोड्स. इसके पास में आपको ये पिंकिश रीजन दिख रहा है. दिस इज medullary sinus. So in medulla there is medullary cords and medullary sinuses. In the cortex there is lymphatic nodules and trabecular sinus, and above the cortex there is subcapsular sinuses. ठीक है. सो दीज आर द की फीचर्स. जो हम 3 से 4 पॉइंट्स की बात कर रहे थे कि आपको उसमें क्या लिखना है, आपको लिखना है दैट इट इज इनक्लोज्ड बाय द कैप्सूल. नाउ दिस कैप्सूल इज बेसिकली dense connective tissue. This capsule sends septas into the parenchyma in the form of trabeculae. Then the parenchyma is divided into cortex and the medullary part. Cortex part has densely arranged lymphocytes in the circular fashion. The center of this lymphatic nodule is lightly stained, and what is happening? Proliferation and maturation of the lymphocytes are taking place. Why it is lightly stained? Because there is less amount. Cells are density of cell is less here in the germinal center. That is why it is lightly stained. Adjacent to this trabeculae, there again this lightish area is representing the trabecular sinus. Now this channel connects this subcapsular sinus with the medullary sinus. Yes. Now from this the lymph will enter here in the medullary sinuses, and जो भी particles, foreign particle, microorganism है वो absorb हो जाते हैं. यहाँ पे जो macrophages है वो act करती है, and ये जो lymphocytes and plasma cells हैं, they will form accordingly, they will activate it and form antibodies. And then these lymph will pass into from the medullary sinus into the efferent vessels and then out of the lymph node. And the blood vessels here, arteries enters and exit, and veins exit through hilus only. Yes. So this is the histology of lymph node. So it has a connective tissue covering in the form of capsule which has, ये जो circular form में दिख रहे हैं ये क्या represent करते हैं? Blood vessels. Yes, जो thin wall होगी that is vein, जो thick wall होगा वो arteries. So it consists of venules and arterioles. Now connective tissue trabeculae arise from the inner surface of capsule and extend through the cortex. Subcapsular sinus is present. Cortex consists of lymphatic nodule. I already told you. Now medulla consists of anastomosing cords of lymphatic tissues. Now what is it is made up of? B cells, macrophages, and plasma cells. Medullary cords interspersed with the medullary sinuses. So these are the sinuses: subcapsular, trabecular, and medullary sinuses. So this is the histology of lymph nodes. So now will you be able to identify the lymph node as soon as you focus? if 10x पे नहीं हो रहा 40x पे करके फटाफट देखिए वापस 10x पे. एग्जाम में हमेशा आपको 10x पे फोकस करके देखना है. तो हमें सिर्फ ये नहीं कि समझ में मतलब पढ़ लिया हाँ देखा नहीं तो भी कहते थे हाँ देख लिया. अपने आप से आप उसी टाइम वापस से किताब को पढ़ के फिर उसको फोकस करो तो बहुत अच्छे से समझ में आता है. सो दिस इज द ओरिजिनल पिक्टोग्राफ. ठीक है, इसमें आपको ट्रेबेक्युलिस भी दिखेंगे, कवरिंग्स भी दिखेंगी. अब मेन इसमें आइडेंटिफिकेशन फीचर क्या है? आपको छोटे छोटे यहाँ पे लाइटली स्टेन्ड एरियाज दिख रहे हैं और यहाँ पे क्या है? ये लाइट पिंक एरिया क्या शो कर रहा है? मेडुलरी साइनस. दिस पार्ट इज कॉर्टेक्स जो डेंसली पैक्ड है एंड दिस इनर पार्ट इज मेडुला ऑफ द कॉर्टेक्स, मेडुला ऑफ द लिम्फ नोड. नाउ द नेक्स्ट इज स्प्लीन. स्प्लीन कहाँ प्रेजेंट होता है? व्हाट इज द लोकेशन ऑफ स्प्लीन? पहले तो व्हाट इज द टाइप ऑफ द ऑर्गन? हमने इससे किसमें पढ़ा है इसको? सेकेंडरी लिम्फॉइड ऑर्गन. नाउ इट इज प्रेजेंट इन द अपर लेफ्ट क्वाड्रेंट. ठीक है, अपर लेफ्ट क्वाड्रेंट द एब्डो रीजन. सो इट इज associated with the 9 to 11th rib with long axis of this passing parallel to the 10th rib. Now size can vary. Spleen filters the blood and helps to remove old RBCs and maintain immunity. It is also known as graveyard of RBCs. So in the lymph node we studied about the cortex and medulla. In this spleen we will study about the red pulp and white pulp. ठीक है, it has a capsule and then there is red pulp and white. So here the key feature is red pulp and white pulp. Germinal center is also there. So red pulp and the white pulp are the main identification features. Now these red pulp and white pulp are separated by a layer of marginal zone. So microscopically किसको नहीं पढ़ना आप जा सकते हैं. हैप्पी न्यू ईयर टू यू इन एडवांस. प्लीज लीव द क्लास. So it is observed that white pulp shows aggregation of lymphocytes and looks basophilic, which is blue in color. Red pulp shows presence of many blood-filled RBCs, and so these are known as blood-filled spaces, sinusoids. This is the summary that we will see in the picture of histology. So spleen, it has a capsule covered by a dense connective tissue layer. Now these again, these capsules sends trabeculae inside into the parenchyma. Now the parenchyma, yes, what happened? Parenchyma has a white pulp and red pulp. White pulp, how you will recognize white pulp? Darkly stained, basophilic. One key feature here of this is central arteriole. This trabecular artery, this will divide and they will form a central arteriole surrounded by. This is surrounded by the layer of lymphoid tissue, which is periarterial lymphatic sheet, which is made up of T cells. I will explain this to you in the diagram also. Then, it also consists of splenic nodules. See, जहाँ पे सेंट्रल आर्टरियोल है उसको जस्ट दे आर सराउंडेड बाय द लेयर ऑफ टी लिम्फोसाइट विच इज नोन एज पैल्स, पेरी आर्टीरियल लिम्फैटिक टिश्यू. एंड जब भी लिम्फोसाइट्स का मैच्योरेशन होगा तो ये आर्टीरियोल जो है ये शिफ्ट हो जाएगा साइड में, उसको बोलेंगे हम एक्सेंट्रिक. पहले तो ये सेंट्रल आर्टरियोल है. व्हेन देयर इज अ फॉर्मेशन मैच्योरेशन of lymphocytes and this arteriole shifts to the one side and it is known as eccentric arteriole. Yes, now the maturation zone is around in the center there is germinal center. So this particular part which do not have this central arteriole and the pals, it is known as splenic nodule or Malpighian corpuscles. Now these two are the features of white pulp. ठीक है, जहाँ पे आप white pulp लिखेंगे, how you recognize white pulp, क्या होता है white pulp? White pulp में densely packed lymphocytes होते हैं. They are densely stained with the basophilic dye and they have a central arteriole. Central arteriole को जो lymphocytes surround करेंगे they are known as pals and जहां पर lymphatic nodules and germinal center है, that part is known as Malpighian corpuscle. Then red pulp is lightly stained, it is more eosinophilic. Then it has again two features, cords of Billroth and splenic sinusoids. Now what are medullary, जैसे आपने cords पढ़ा उसमें क्या थे, B cells, macrophages, plasma cells. Here cords of Billroth is made up of RBCs, then again macrophages and lymphocytes. जहाँ पे cords हैं, they basically have these cells only, and additional क्या है, RBCs. Splenic sinusoids, these are the blood filled spaces which have endothelial cells known as staph cells, and they are filled with the RBCs. Marginal zone is basically differentiating white pulp from the red pulp. तो इन दोनों के बीच में जो cells होंगे marginal zone में, वो differentiate करेंगे white pulp को red pulp से. Now this is the diagram of spleen. अब ये मत कहना कि lymph node से similar है. क्यों नहीं है similar? ट्रैबेक्यूले पूरे extension में नीचे नहीं आ रहे हैं. ट्रैबेक्यूले थोड़े से बस कुछ देर तक ही इन्वेजिनेट करेंगे. So the short trabecules, which are the septas only sent by the connective tissue. So this outer connective, dense connective tissue layer, septas are there which are in the form of trabeculae. Now the two important feature of the spleen are white pulp and red pulp. Now the task is to recognize what is white pulp and what is how to recognize here. तो white pulp क्या होगा, basophilic. तो बेसोफिलिक जो भी आपको यहाँ नजर आ रहा है क्लस्टर में, दैट इज वाइट पल्प. ये क्या है, लिम्फोसाइट्स ही है. और वो जो लिम्फोइटिक नोड्यूल्स हमने पढ़ा जहाँ पे वो सर्कुलर फॉर्म में अरेंज होंगे और सेंटर में लाइटली स्टेन रीजन होगा, दैट इज नोन एज जर्मिनल सेंटर. वो कहाँ प्रेजेंट होगा, वाइट पल्प में. और इसका इंपॉर्टेंट की फीचर क्या बताया था मैंने, सेंट्रल आर्टीरियोल. जब भी ये प्रोलिफरेशन होगा तो सेंट्रल आर्टीरियोल it will be shifted to the side. Now the lymphocytes which are just surrounding this central arteriole, they are known as periarterial lymphatic tissue or pals. So what are the parts of this white pulp? We have pals surrounding the central arteriole and Malpighian corpuscle. Okay, now how to recognize? Now this red pulp is scattered all over this spleen. Yes, here you will find eosinophilic areas which is red pulp. Now red pulp, it has sinusoids which are blood filled spaces, so that will appear red in color, and cords of lymphocytes and macrophages that will appear little basophilic. Okay, now these parts, these are the parts of the connective tissue only, septas only, so don't confuse them with the this red pulp. These are not red pulp. Red pulp ये जो इधर के एरिया में प्रेजेंट है यही है. तो व्हाइट पल्प को और रेड पल्प को जो सेपरेट कर रहा है वो कौन सा जोन होगा, मार्जिनल जोन. ठीक है, तो ये कंप्लीट होती है हमारी स्प्लीन की स्लाइड. सो व्हाट आर द कैरेक्टरिस्टिक फीचर्स हियर इन द माइक्रोएनाटॉमी ऑफ स्प्लीन? व्हाट यू विल से, there are white pulps and the red pulps. White pulp are more basophilic, red pulp are eosinophilic. White pulp has central arterioles surrounded by the pals, periarterial lymphatic tissue. Then there is lymphatic nodule, there is a central germinal center, and because of the proliferation the central arteriole is shifted to one side and it is now known as eccentric arteriole. Then all around this there is scattered red pulp. तो red pulp में how to recognize splenic cords, जो कि ब्लू स्टेन जो होंगी इस पूरे एरिया में, कॉर्ड्स की तरह ब्लू स्टेन्स जो होंगी, दैट विल बी रेड, दैट विल बी दी स्प्लिनिक कॉर्ड्स. एंड जो रेडिश स्टेन ले रहा है दैट इज दी साइनोसॉइड्स हैविंग दी ब्लड आरबीसीज एंड मैक्रोफेजेस. दिस इज द हैंड ड्रॉन डायग्राम ऑफ spleen. Now the next one is thymus. Development of thymus, have you read? किससे डेवलप होता है? डेवलपमेंट इसका इंपॉर्टेंट है, एम्ब्रियोलॉजी में आपको पढ़ा दिया जाएगा. इट डेवलप्स फ्रॉम द थर्ड pharyngeal pouch. Then it proliferates and it grows till the age of puberty and then degenerates. तो फंक्शन क्या है, अभी हमने पढ़ा था, इट इज प्राइमरी लिम्फोइड ऑर्गन. सो एजुकेशन एंड मैच्योरेशन ऑफ लिम्फोसाइट्स, मतलब एजुकेशन का मतलब क्या है, एजुकेशन एंड मैच्योरेशन डिफरेंशिएट होगा लिम्फोसाइट्स एंड एजुकेशन का मतलब पर्टिकुलर सरफेस मॉलिक्यूल पे पर्टिकुलर जो वो अटैच होगा ना, देयर इज अ मेजर हिस्टो compatibility complex molecule is there, so it recognize this MHC and get attached to the and destroy the foreign or microorganism. It to provide isolated environment for the lymphocyte maturation, secretion of thymopoietin by epithelio-reticular cells. Thymopoietin stimulates T cell production. Thymus is essential for the development of immunity. After puberty, thymus gets atrophied. So basically it is related to the maturations of differentiation of T lymphocytes. So capsule is present here. Now there is a different picture as we have studied in the lymphocyte, this lymph node and spleen. In the thymus, yes. Now in the thymus, it is surrounded by the capsule. Now these septas, they divide the capsule in numerous lobules. There are different lobules over here. हर एक सेप्टा जो है वो क्या कर रहा है, सेपरेट कर रहा है टिश्यूज को अलग अलग लोब्यूल्स के अंदर, और ये लोब्यूल्स जो है मेडुला में जाके आपस में, इनका मेडुला इज कनेक्टेड पर इनका सबका कॉर्टेक्स जो है वो सेपरेटेड है. तो कुछ इस तरीके से पिक्चर आपको दिखेगी इसकी थाइमस की, जिसके अंदर इट्स कवर्ड बाय द capsule. It sends the septa trabecular inside and divides the tissue in different lobules. Again same thing is there. There is lymphatic nodules. There is a light stain zone which is germinal center. The main characteristic feature of thymus here is presence of Hassel's corpuscle. वाइवा में ये आंसर दे दिया, so will get the highest marks. कोई आंसर नहीं देता इतना बताने के बाद में भी कि ये है क्या. So basically capsule is there, trabecular are sent inside and which divides the thymus into different lobules. There is a outer dark zone which is cortex and inner zone is medulla. Cortex contain densely packed lymphocytes that do not have lymphatic nodules. Medulla have few lymphocytes but more epithelial reticular cells. Hassel's corpuscles are there. So this one lobule, it has outer densely part which is known as cortex. Then जो ये reddish part है inside the medulla, this is Hassel's corpuscle. Now what are these Hassel corpuscle? These are the type 6 epithelio-reticular cells. Epithelio-reticular cells are present here in the thymus. They act as supporting cells. ये 6 types के होते हैं, type 1, type 2, type 3, type 4 and type 5 and type 6. So the Hassel's corpuscles is type 6 epithelio-reticular cell and it secretes interleukin 4 and interleukin 6, helps in thymocyte maturation, and they also produce thymosin and thymopoietin hormones. लिख लो, याद तो रहेगा नहीं. ये क्या है Hassel corpuscle? Please write down because you do not answer in the exams. These are the small rounded or ovoid structures which are present in the medulla. Medulla क्या है? Inside lightly stained zone which has this ovoid hyaline mass which consists of concentrically arranged epithelio-reticular cells. What are the function of these cells? They secrete interleukin 4 and 6 and they produce thymosin and thymoprotein hormones which helps in the maturation of thymus. Thymocytes are the lymphocytes which are present in the thymus. So thymocyte naam de diya inko. So main differentiating feature of this is Hassel's corpuscles and presence of the lobules which are not found in the spleen and lymph nodes. Different lobules are there. अभी जो center में reddish area दिख रहा है, these are scattered Hassel's corpuscles. So you have to draw the outer darkly stained area, inner medulla and Hassel's corpuscles. And if you're done with the Hassel's corpuscles, you will get the full marks. Yes? And you must know the background story of the thymus. We have discussed. What does it do? What are the functions of thymus? Now the last tonsils. Tonsils you will study detail in the gross anatomy. Here we will study the microanatomy of tonsils. Please be quiet. This is the last slide. Then you can go away with whatever you want to do. Tonsils are collections of lymphoid tissues near the junction of oral and the nasal cavity with the pharynx. Aggregations of tonsils form Walder's ring. Now, what are the types of the tonsils that are there in the Walder's ring? Pharyngeal tonsil, tubal tonsil, palatine tonsil, lingual tonsils. Basically, what are tonsils? These are the lymphoid collections of the lymphoid tissues which are present near the junction of the oral cavity and nasal cavity with the pharynx. Now, they are present in a circular, they form a ring. Different tonsils, they form a ring which is known as Walder's ring. Because of their, when we connect these, there will be formation of a circular pattern which is known as Walder's ring. So Walder's rings is formed by pharyngeal tonsil, tubal tonsil, palatine tonsil and lingual tonsil. Palatine tonsil is the largest tonsil in the group of tonsils. Now what is the location of palatine tonsils? I told you should know the background story. आप जो भी histology पढ़ेंगे पहले उसके बारे में आपको पता होना चाहिए कि actually है क्या. So this is a collection of the lymphoid tissues. Now where the collection of the lymphoid tissues? So it is present on either side of oropharyngeal isthmus. It's an opening between the oral cavity and pharynx. So दोनों ये कहाँ present होंगे? On the right and the left side of the oropharyngeal isthmus between palatoglossal arch and palatopharyngeal arches. It is present between palatoglossal and palatopharyngeal arches. ये क्या है? That you will study in the gross anatomy. If you open the mouth, there are two arches formed behind. ठीक है, एक तो आपका ऊपर से टंग की तरफ आ रहा है, that is palatoglossal arch, और एक आपका कहाँ से बन रहा है? From the uvula, soft palate से नीचे की तरफ. So these two arches, in between they have this triangular region, they have presence of palatine tonsil. So it is present on either side of oropharyngeal tonsils. You put a star mark if you do not understand now, because it will be covered later. It is, you just write the location between palatoglossal arch and palatopharyngeal arches of the soft palate. Now tonsils are a part of mucosa associated lymphoid tissue mold that we have discussed. Mucosa associated lymphoid tissues. Only lateral part of this is covered by the capsule. So this is the location. See, this is nasal cavity, oral cavity. Then this is the part of the hard palate. This is the soft palate. Now there are two folds over here. The one which is coming towards the tongue, this is palatoglossal arch, and the one which is going downwards towards the pharynx is palatopharyngeal arch. So here in this triangular region, this is the location of palatine tonsil, this one. Now these are the three important points that you have to write. This is the most easiest slide out of all that we have studied, palatine tonsil. First is the lining epithelium. You will get half of the marks here only. Then tonsillar crypts. This tonsillar crypts is the word which external want to hear and want to see in your answer sheets. Lymphatic follicles. So in the lining epithelium, what will be the epithelium? You have read this, I think, in the examples also. It is see, just oral cavity. Here there is non-keratinized stratified squamous. So it is present near to the oral cavity only. It is covered by non-keratinized stratified squamous epithelium. Now this epithelium invaginates inside and form tonsillar crypts. Yes, again there is tonsillar crypts. These are lined by non-keratinized stratified squamous epithelium. Then the lymphoid tissues are there, which are aggregation of lymphatic follicles supported by the reticular fibers. All these lymphoid tissues, they have reticular cells in the reticular fibers. Reticular fibers are type 3 collagen fibers. They form supporting network for the lymphoid tissue. Now boundary between the epithelium and lymphoid tissue is not clear. There is no demarcation there. Functions. productions of activated lymphocytes, antigen presentation, and development of immune response. Because what type of organ it is? Secondary lymphoid organ. This is the summary of the tonsils. Now this is the microanatomy. So outer layer is the epithelium, which is made up of yes, non-keratinized stratified squamous. Now, this epithelium is invaginating inside, and these are forming crypts, which are known as tonsillar crypts. Then again here near the epithelium, you will see arrangement of the lymphatic follicles that we have studied: outer, darker, densely packed lymphocytes, and inner light zone, which is known as germinal center. This you will see all around here. Then below you will see laterally there are capsules and skeletal muscles present below these layers. But you have to answer what? Epithelium, tonsillar crypts, and lymphatic follicles, which are arranged below the epithelium and tonsillar crypts. So this one is the most easiest. So let us quickly revise the picture only. Only picture, nothing else, no function. Function now you go and read from the book. So we will start from again from the last to the first one. This is the slide of tonsils. Tonsil, what type of tissue it is? Secondary lymphoid organ. Then what are the key features here in the histology? It has non-keratinized stratified squamous epithelium. Then it has tonsillar crypts. The tonsillar crypts क्या है? Basically invagination of the epithelium of the tonsils. These are again lined by the non-keratinized stratified squamous epithelium. Now below epithelium there are presence of lymphatic, circularly arranged lymphatic follicles. Then this is a slide of thymus. It is covered by the capsules. Capsules send septa inside into the parenchyma and divides the organ into different lobules. Now these lobules have the outer darkly stained zone, which is known as cortex, and inner lightly stained zone is medulla. जहाँ light stain है मतलब वहाँ पे there is less amount of lymphocytes. Yes, that is why it is lightly stained. Then what is the key feature here? Most important, Hassel's corpuscle. And what are Hassel's corpuscles? These are the collections of type 6 epithelio-reticular cells. So this is the slide of spleen. Key feature here is red pulp and white pulp. How to recognize white pulp? Central arteriole. Yes, and central arteriole surrounded by, yes, basophilic. Yes, basophilic, circularly arranged. Yes, you are right. But then important feature is that they are present near central arteriole. The diffuse layer is sparse and it is also associated with, yes, splenic corpuscle. Then inside we have scattered red pulp, which has cords of Billroth. They are known as cords of Billroth, which are again lymphocytes, plasma cells, macrophages, and they are surrounded by sinusoids. Last is lymph node. Lymph node, again there is a dense connective tissue layer which sets trabeculae inside to the parenchyma. Below the capsule there is, which sinus is there? Subcapsular sinuses. Then below the subcapsular sinuses, lymphatic nodule, which has outer mature zone and inner central lightly stained zone, is germinal center. Then adjacent to the trabecular, which sinus is there? Trabecular sinus. Now this trabecular sinus connects subcapsular sinus to the medullary sinus. What opens in this subcapsular sinus? Afferent vessel. Then here in the medulla we have medullary cords of again B lymphocytes, plasma cells, macrophages, and they are surrounded by medullary sinuses. Trabeculae opens into medullary sinus, and from the medullary sinus it continues into the efferent. So outer one is the cortex, inner one is medulla, and again cortex has two types: nodular cortex and paracortex. Nodular cortex has B lymphocytes, paracortex has T lymphocytes. Medullary cords have predominantly B lymphocytes. CR